All right. Hello, everyone. If I can call your attention back, we're going to start into an interesting discussion. There's been so much fruitful that's been discussed here already, and I do have the sort of feeling that when we do dive into this, we will probably barely scratch the surface. I was going to get things kicked off with a particular question, which I'm actually just going to open to the entire panel, which is a twofold question. How can planetary science help us understand the context of potential life on exoplanets? And could exoplanets help us understand important things about our own solar system? So does anybody want to start with that?
Sure.
All right.
I'll say a little bit. Definitely, I think we can learn so much from our solar system. As I said, it's our main data laboratory that we can get some real data about origins, life, where can it exist. And I think that's my most exciting trajectory, to go to exoplanets. But I think exoplanets can offer something as well. If we can see differences in atmospheres that seem to not be just from geology or physics alone, and we start to see signs that we don't understand in unknown chemistry, or even if we see known chemistry and can see more about where the origin of life can exist and in what sorts of statistical ways it can exist and in other systems, I think that'll also help our own understanding of where the origin of life can take hold. So I think hopefully that's a long-term vision. I don't think I'll necessarily live to see that, but eventually I hope it'll help us understand.
Yeah, it's very complementary in that in our solar system we do have a wide array of worlds and worlds we can get to get some ground truth, if you will, or atmospheric truth. But it's one data point of a solar system, and being able to see the wide variety of solar systems, we see with the exoplanet systems is really important to understand aspects of formation and evolution, and evolutions in particular of habitable zones. So I think we need kind of both approaches.
Great.
I was going to say that my favorite transmission spectrum, so I just had this, sale pitch of transmission spectra, is actually the Cassini transmission spectrum of Titan. The reason why that's true is because that backlit observation of Titan, is dependent upon color information about how the atmosphere transmits light, which then it's really Ty Robinson who led this study, taking the light alone and then extracting information about the hazes in Venus. What was so satisfying about that to someone who studies exoplanets was that those measurements were confirmed in situ. So like the particular particle size that Ty and his team inferred from that transmission spectrum was confirmed by the particles that Huygens tasted, I think, as it was like plummeting to the surface of Titan. And then the size of the features was validated by the measured surface gravity, but the speed at which Huygens plummeted, it was just deeply satisfying, and also ground truthed transmission spectroscopy as a method for understanding planets in detail.
Great. Thank you. I think that leads kind of naturally to, from talking about chemistry of life and what we can understand about that, to some of the potential for Titan and for the clouds of Venus and surface of Mars and various other objects in terms of what it can tell us about prebiotic chemistry. Maybe you can't start life on Titan. Maybe you can't start life in an asteroid or in some sort of planetesimal body, but it does seem like a lot of prebiotic chemistry is happening. How far can that prebiotic chemistry really go, do you think? And both in terms of how far can it go toward life, and how much could it tell us about the potential for understanding better the possibilities of prebiotic chemistry on the surface of early Earth? This is for all of you, but Sarah,
I'll start. And I'm not an organic chemist, I'm a mineralogist, and frankly, this is something I would like to know. So, we know that, I think Bennu has taught us that it's actually easy to make these very complicated organic molecules. And we saw this from the Rosetta mission as well to Comet 67P, that there's this whole zoo of organic molecules that are produced. But one thing I'd like to understand, maybe from you, Paul, or the audience, is why we can't go from that step to the moment of creating life. There seems to be some magical thing that at least I don't understand. I'd like to better understand from other people here.
Yeah. Well, I would like to derail the conversation a little bit, if I may.
That's fine.
We were told to be a little bit controversial, so here I am for it. My point, which I quickly flashed in the slide in my presentation, was that the premise of the conference is wrong. We are actually not interested in life or consciousness when we are looking up to the stars. We are interested in interesting things in general, and we are interested in things which are dangerous. As an example, for example, we had a COVID pandemic quite recently, and we still don't know if viruses are life or not. The debate is still raging for over a century, right? There are more important things to know about the system than whether to call it life or not. The same goes on the other side of the scale. If we see a swarm of spacecrafts coming out to us, we are not going to worry if they are conscious or if they are alive. It can be just a swarm of self-replicated von Neumann probes, which don't have consciousness. They are self-replicating without evolution, so they are not live, but we still would like to know about them, right? Yeah. So no consciousness, no life, we still want to know about it. And finally, the Venus. Speaking about controversies, phosphine, how you generate it, right? There are some debates, but still parts per million concentration of phosphine was a billion, yeah. Very low concentration. What I just showed, we need 20 grams of highly absorbing organic material in clouds of Venus to make it as dark as it is. So, yeah, and again, we didn't start it with the premise we are looking for some life, or consciousness, in this case, it's irrelevant. We started with the premise, hey, let's look into it, and then let the data inform us. So I think the approach should be we first find it, and then we decide or let philosophers debate whether it's life or not. I don't care if it's dangerous, is the more.
I sense that there will be a few responses to this.
Actually, I was just thinking in terms of the how far the chemistry can go.
Oh, yeah.
One of the reasons we want to explore is to see to see what has happened and to get that data to interpret. But if it hasn't progressed as far as we might expect based on laboratory experiments, that's also important. If you put all the ingredients together, in a nice warm pool of liquid water on the surface of Titan and let it mix for a few thousand years and nothing happens, that's important to know as well, to be able to understand what the factors are that are the most important. So even the negative result is going to be really fundamental to pushing us toward the next steps.
I just want to say that actually I completely disagree with what you just said. I completely disagree that we're not interested in life. I think we are interested in life and consciousness, even if it's not dangerous to us, because it tells us something about ourselves, and we can learn about ourselves by looking at aliens.
So if you find von Neumann probes which are not live, you would not be interested?
Of course, but it's not an either or. Yeah, right. It's. I'm interested in that. I'm interested in also what's the distribution of life, and if it's weird and different, or if it's the same. If life's biochemistry is highly constrained or not. How often does consciousness arise or not? Also, if self-replicating probes come, also interested. I see this as just we're curious, and I am still interested in the definition of life.
I agree with the part that, for example, with the viruses, we still argue about it, right? Mm-hmm. And so my point is that there are more interesting things to know, and I agree with the part that it tells us about ourselves. So then some person says that virus is not life. It tells me something about their, approach to studying it. It doesn't tell me anything about the virus. So some people believe that viruses are life. Some people don't believe that. But that was my point. I think I made it clear. Some controversy to start with, but
moving a little bit to a sort of question that would be less ambiguous. I'm not sure, Craig, if you are able to speak up here, but I was really curious in terms of how remote sequencing would work and how flexible it would be. You said it would be a huge shock if you found life as we don't know it. How different from DNA could you really get before this whole sequencing project would fail? And what's the hope of finding something genuinely different through your sort of teleportation technique?
The chemistry of sequencing is very dependent on the chemistry of RNA, DNA, so we could easily detect either a it's a different molecule, the quaternary space alone. Only be able to detect, but there's just with the spectrometry, you're looking at the 260 wavelength of the bond that joins DNA and RNA pushes 280 that detects bookkeep bonds. There's ways to simply look for both. But DNA sequencing and RNA sequencing are dependent on hemispheres, we understand.
All right, I think that makes a lot of sense. So I did actually have a question a bit about exoplanets, something that I always kind of wonder, keeps me up at night a little bit. We find a signal on Venus, maybe, and maybe it's something interesting as a biosignature, and we can have all these great questions. We can send a probe there and we can find out. We can't do that for any exoplanet. And so we're just going to keep getting ambiguous signal after ambiguous signal. The two-part question to this is, what sort of future instruments do you think are really going to help to get a sort of statistically robust, and these could be future observatories that will likely exist or future observatories that you personally are dreaming about? And then the other question is, how do you communicate that responsibly to the public without misleading them?
Oh, yeah.
So,
I think about this a lot. I think this whole, we've learned a lot, from having these controversies of Venus and phosphine. And I'm sure we'll have the controversy, of course, of dimethyl sulfide. And it's made me a lot more humble. I think when I was a bushy, bright-eyed grad student, I was like, we can detect life on an exoplanet sometime in my lifetime. And then seeing how difficult it is, even in our own solar system, when we have really good observations. And, it made me a lot more skeptical that we're going to actually get any answer for exoplanets that we're going to be really satisfied with, because we can't go there and we can't ground truth it. And even where we can have really good observations, it's still difficult. So I think it's made me a lot more skeptical that we're going to actually get robust signs of life. I think we're going to be debating it a lot. And that's part of the scientific process. And as far as communicating it, I'm of two minds, because one is, I think we need to, when we convey these findings to the public, we need to convey them with the proper uncertainty. We need to say that any sort of controversial claim is going to bring a lot of eyes to it. And you're going to have real analysis of the data. You're going to have people disagree with that. And that also, I hope, is conveyed to the public, because I think that we're in this unfortunate period of our time in the world where people are mistrusting experts, mistrusting science. And what they're seeing is the scientific process unfold. Someone makes a strong claim. Other people say, oh, but did you consider this? Or, well, this pipeline is maybe not so good there. Or, actually, we can just fit a flat line to this data. There's so much that comes from that scientific debate. And so we need to do a better job conveying that process, because that is science. That's why it's successful. That's why it's successful at knowing things, because it's falsifiable and that we can actually change our mind and disagree. So, and then the second part, life mission, totally. I do think that's going to be one of our better ways. Also, if I had like an infinite budget, I would put, a high resolution spectrograph on the moon or something like that, and then be able to pick out the minor species.
I think another resource we have that we don't always take full advantage of is, the spacecraft we have in our solar system and turning them back to look at Earth. And would those spacecraft detect, be able to detect and definitively detect evidence of biology here on Earth where we know it exists? So I think we have some opportunities that maybe we haven't been able to take full advantage of to do that comparison as well.
Right. Sarah Ballard, do you have anything that you'd like to add to this?
I was thinking more about like the existential aspect of that question. It's very similar to a question I posed at a similar meeting probably a decade ago. I was the question asker, and I asked with respect to the public and with respect to ourselves as well, ultimately, what does it mean to be satisfied with a finding, especially a finding of this nature? And so I think I said at the time, would we have to hold a bug at the end of the day in order to feel satisfied? But I'm speaking now to a human experience of knowledge. And so but the extent to which anything can be known ultimately is probabilistic. I'll have to make my peace with that, Stuff. Yeah, and which is sort of a very human question. Yeah. That's my response. Yeah.
Thank you.
Just to quickly say, I think also there's maybe a different standard of proof at the moment for our solar system and exoplanets. If you look at Mars, for example, so I think there was a time long ago when it was thought that if there was water on Mars, then it must be in the Goldilocks zone, and then it would be, there might have life there. And we now know there's water, there's mudstones, there's methane, there's dimethyl sulfide, there's all of these things that are potentially called a biomarker on exoplanets, but there's a feeling that we actually need to, get up physically our hands on life to be able to really be able to prove it's there.
Yeah, I completely agree with this point. Whether the life is the one as Craig can sequence, or if it's a little bit different, we still want to get our hands onto it. The remote observations of dimethyl sulfide would, as long as there is a chance that the molecule is produced abiotically, it's going to be very hard to get from series of unambiguous detections to claim that, yes, we found life, whatever you decide to define it. The difference between searching for life on Mars, where we can actually send physical probes and analyze the sequences, it's a much different claim than observation of remote planets. One more thing, there's this urgency I was talking about in my talk. I think we should prioritize a little bit when it comes to search for life. That's the thing I'm advocating for, obviously. We have centuries to observe exoplanets. We have only a decade to observe pristine Mars. So that's my point.
All right. I think. Oh, please.
If we're looking for life in the oceans, they are off by about 6 orders of magnitude. And you can look at the crystal clear water, but unless you're a high-power microscope today, or through the lens of DMA sequencing, you would assume there's no life there. But it turns out every milliliter seawater has over the million microbial samples numbered in the virus. They're just not part of our visual acuity, so we assume they did things as recently as a decade and a half, building through DNA sequencing the oceans, that we showed the absolute abundance of life by discovering tens of millions of new species. So science tends to be very myopic, EP threat for us. So trying to find it elsewhere becomes a bigger challenge.
I think that that's an incredibly profound insight from life in a very alien environment. Connects with what we've seen about the genuinely alien nature of certain kinds of small animals and octopi and various other things we got to hear about before. I think that that's a perfect transition, actually, to open to audience questions. So I can't see any of you pretty much, so I will trust the person walking around with a microphone to make decisions.
Hey, yeah, hello, I'm here. So yeah, thank you. Wonderful session, wonderful conference. I'd like to take up Jan's kind question. You know, Jan, I think you were rightfully basically saying, hey, aren't we just with consciousness and life, aren't we just scratching the itch of curiosity? And aren't there more important things we should actually be doing? And I think we have some nice answers and good answers for life. And here's my answer for consciousness. And what I'd like to say is that there's basically a huge misunderstanding. And the misunderstanding is consciousness science is not about getting knowledge only. It's about making change. It's about solving big parts of medicine. It's about solving chronic pain, solving nociplastic pain, solving a huge amount of psychological conditions. It's about understanding the new systems we have on Earth now, AI. It's about making new take. So it's not an enterprise of gathering understanding. It's an enterprise of coming up with change. So I would argue it's not even in the ballpark of things that you could direct that criticism at. That said, I appreciate the confrontational tone, and I think that's exactly what we need.
And Yanni certainly should have an opportunity to respond.
Yeah, thank you. Well, the point is, for example, with the octopus example, if we find aliens who are looking or have similar behavior to octopus, I think it's more important to know whether they are intelligent than if they have consciousness.
Isn't solving pain more important than even what you just said? You know, is it?
Well, as long as we are talking about looking out in the universe, if we are looking for medicine, then yes, of course, that's a different point. I think that this conference has different topic.
Thank you. Several speakers talked about contamination, and I like the question of why life didn't emerge on Bennu. And I would like to ask, how hard would it be to contaminate the asteroid T with some form of life? Would it be very difficult? Does life actually like it, on Bennu?
How difficult would it be to contaminate the asteroid T? Is that the question? Yeah. Incredibly easy. Yeah. So especially with, I've learned that organic molecules are everywhere, and they can get into all sorts of places no matter how well you try to contain them. So for Bennu, it's being kept in a pristine nitrogen-rich atmosphere, just taken out for analyses when necessary. And the organic work that I presented was done with blanks alongside them. So they're fairly confident that what they're seeing is the true signature rather than any contamination. But yeah, of course, that's the fantastic advantage of having an asteroid sample return mission rather than meteorites, which are basically all contaminated, no matter how careful we are with our curation of them.
Just a very quick follow-up from my own curiosity. Is there a risk that by landing there and taking a sample, if we weren't very careful about how we've cleaned that instrument, have we now contaminated that asteroid already?
Yeah, sure. That is a concern. So I'm sure there's this whole subject of planetary protection, which includes both trying to protect the Earth from alien life, but also trying to protect other planetary bodies from contamination on Earth. And I'm sure, so every space mission has to go through a process to show that contamination is not going to hopefully cause too many problems. I guess the Dragonfly mission might have some thoughts about planetary protection. Right.
Actually, Titan is a category 2 in that the ocean is very deep. It's about 100-kilometer ice crust. And so in terms of planetary protection, we absolutely have to pay attention to that. But the requirements that are levied from the planetary protection community aren't as stringent as it would be, for example, going to Enceladus, or if you were landing on the surface of Europa, where you might actually have a higher chance of contaminating a present-day liquid environment. With Dragonfly, we can't create a warm enough oasis around the lander itself to really do any forward contamination of Titan. We do have to pay a lot of attention to contamination control, because the last thing we want to do is get to Titan and pick up a sample with the mass spectrometer and detect something that we brought with us from her. So there's still a lot of cleanliness controls, and planetary protection is of course something that's taken very seriously across the solar system in terms of that forward contamination and preventing it.
Great.
Back here, Paul.
Oh, great. Oh, okay. I see a hand waving at least.
Hi all. This is a question probably for the Sarahs. You talked about how we're all just going to have to come to terms with this idea of just being, it's just going to be like a probabilistic sort of like if we detect life, not like a sure thing. So I'm curious, in your expertise, what would be the combination of molecules? Because you said it's not just going to be one single thing. What would be the combination of molecules that would give you the most confidence and the highest probability to say, yes, that is the most likely thing that we can point to is life on an exoplanet somewhere?
Yeah, I mean, I hate to say it. I do think it's the combination of Earth biosignatures that would, I think be the hardest. It's certainly been the hardest to find false positives for. A strong oxygen or ozone plus methane, we just have not produced abiotically, like with water and CO2. Not saying we can't break that false positive. Maybe there are ways, especially there's one paper that shows you could be confused by a moon in the system, for example, and get that a false positive of oxygen and methane. But in the atmosphere itself, if we're convinced it's coming from the atmosphere, right now this seems very difficult to do because the lifetimes of those. I just wouldn't be convinced by a single molecule. So that kind of rules out a number of these like more minor biosignatures that we're talking about, just because, say on a different planet, the formation mechanisms there, the chemistry could be very different. So I do think we're going to be looking at a combination, and I hope that we can eventually look more agnostically for non-earthy biosignatures. But right now, that is certainly what we could understand best, and probably that's when I would really open the bottle of champagne. Sure.
I was thinking, sort of trying to place this conference in time. So I was talking about what I would make peace with. I don't know if I'll be around for what I think will be the next level of satisfaction, which I would say would be associated with direct imaging of small planets. And so that's talking about optical interferometry on scales that we can't really conceive of them now. I saw a talk once where someone asked a speaker, well, how much would that cost? And he just laughed. There was no answer. So just fine. I don't think he even thought about it. And yet to imagine seeing a picture, right? And then you're talking about maybe the relative albedos of different species on the surface. Does it look like lichen, for example? So I would call it another level of satisfaction. Is it like complete satisfaction to the extent of holding as an organism? No, but there are levels. I don't know if we'll be alive for that. What I posited just now, and also it will depend, on a lot of choices that we make in terms of funding and so on.
For sure.
We should move on to the next question.
Is it working? Okay, good. Betül Kaçar from University of Wisconsin-Madison. So we started this session through a very inspirational opening about the importance of big thinking in education and whether we are accessing that or whether we're giving students this sort of inspiration at today's education system. And I will link that to what Jan said about how this is an all hands-on-deck problem. Yet we rarely see the hands of biologists in the search for biology in the universe, which I find quite daunting. So my question, even though I'm looking at your faces in the audience, is for Craig Venner, as the sort of one of the few biologists that also had the pleasure to listen to an astrobiology life search meeting. Why do you think that biology has been sort of staying outside of search for biology in universe studies? Is it a disciplinary problem? And how do we fix that? Because I think there's a tremendous amount of potential for especially synthetic biology in partnership with astronomers and geochemists and planetary scientists, and for the contribution of search for study of life in the universe, because ultimately it will come down to biologists who put the centuries of work on this planet identifying what biology is capable of doing, and convincing that group ultimately is going to what this come down to. So what do you think about the hurdles and changes that we should also think about catalyzing in order to push this forward? Thank you. And of course, Craig and anyone else who wants to answer is welcome.
It's a complex question, but I would be satisfied if I saw evidence of DNA molecules or RNA. RNA can be self-replicating, so I think that would convince people who think that RNA was a virgin of life. But I also think there were tens of thousands, if not tens of millions, origins of life on this planet. My view, you get the right chemicals together and life will happen in the right environment. And the diversity that we have life and find on this planet when we look for it is pretty humbling from a field that started with a human-centric view of life. We're sort of the Porsche example of life from many aspects of things that can solve, survive in extreme conditions, but it's still difficult for people to see and identify life here without the tools of science. So without the tools of science, changing and improving is going to be difficult to see in elsewhere. But we should be looking first and foremost for microbial life, because you're not going to get higher life without viruses and microbial.
Can I have a
please
comment? Craig, two things. One, would you like to team up with us? We'll provide a concentration. You can do the analysis. That is the first part. Second part, how likely you think it's that there's going to be genetic drift in actually chemistry of the nucleobases. For example, we know that we have some viruses that have amino adenine instead of adenine, and you cannot find them with classical PCR. So if you can touch up on that.
People have made a number of synthetic components now of life, the things that had not occurred naturally, so we can expand the repertoire, but there are minor changes compared to the basic molecules. We're having a team up with NMA, find a real sample for us to look at.
Okay.
Hi, Chris Temby from the Agnostic Life Finding Association. Hopefully this is a fun one, but and
this will probably be the last.
This sounds good. Okay.
One more.
Okay. I'll be quick. Talking about life as we don't know it, is there any hope to try to understand, what life could be on rogue planets? Or for example, there's a interesting philosophical conversation of would a species or an intelligent life form that's in the ocean of Europa be able to actually consider itself in the cosmos, given it has no perspective of the stars? But in a more practical sense of the question, could we ever hope to find any life forms on a rogue planet, or is that strictly the realm of science fiction?
What a great question. Who wants to respond?
Yeah, it's hard, right? Because right now we're limited to trying to see something with an atmosphere and a global biosphere. So that's what I was saying, just because a planet's habitable doesn't mean that it's useful for us. So it could be theoretically habitable. There could be Europas around other systems that are very habitable environments. And if I were living in that system, I would want to go there and look for life, which is what I think we should do in our solar system. But as exoplanets, they're just useless to us right now. And I can't see a way around that. Same with rogue planets. It's hard to imagine, like, how are we going to, see a biosignature unless the rogue planet is beaming at us like a prime number sequence, and then great. But otherwise, it's so hard. So we're really hamstrung in our efforts to kind of look where the light is shining. It's the old adage of someone lost their keys and the person's only looking where the lamp is, and it's like, well, this is where the light is, and the keys might be somewhere else. And I think we're in that position, unfortunately, where there's probably life that could exist in all sorts of different environments that we don't even understand. And certainly some of these environments are very Earth habitable, like Europa is another system, classically habitable, that known life could exist there. Yet it's just as an exoplanet, really hard to think how we would ever be able to detect that. So there's kind of this limit of where do I think life could be versus where do I think we're going to detect it. And then hopefully, over time, we'll be able to expand our efforts.
I think with just one step sideways, that question becomes like much more answerable and, in my opinion, much more tasty, which is like, so if you're thinking about life without the benefit of a main sequence star, fine. Just look around white dwarfs. I think some of the most interesting astrobiology relevant, like geology, is from white dwarf pollution. But on top of that, you could potentially have second generations of the evolution of life around white dwarfs, which would receive the benefit of additional enrichment from the things which would have fused inside the star during the nominal main sequence lifetime. So, like, I heard rogue planets, but I was like, we should be talking about white dwarf planets. It should have its own whole session.
No, I think that's absolutely awesome. White dwarfs are amazing. I was actually going to follow up on Sarah Ruckheimer's statement and connect it with a question from the audience, which is about the prime number sequence, right? That's much less ambiguous. Given the fact that that's much less ambiguous than this constantly ambiguous, controversial arguing about trace amounts of sulfur or phosphorus containing gases or whatever else, would this... here's the real controversial way to say it. Would this mean that we should be putting proportionately more money into SETI rather than basically rolling dice on something that's not going to convince everybody ever?
Yeah, you just made my point. If you are looking for a sequence of prime numbers, you are not looking for consciousness or life, you are looking for intelligence.
I mean, I think, again, I would say...
I'd make the leap personally, but...
Absolutely, we should be funding, SETI, Breakthrough Listen, these sorts of endeavors. And we also just want to know how planets work. The other thing is we're not necessarily only in this game to find life. We want to find what is the wild diversity of exoplanets, worlds that are totally unlike our own. And we'll learn really interesting things there. And I think that's the wonderful thing about science. We're not really sure what we'll find, but it'll be exciting along the way. And doing these laboratory experiments, of different sorts of alternative chemistries is also interesting. But I do think it's like, yes, and hopefully funding dependent.
Yeah, we not only do we not necessarily know what we find, we don't know which approaches are going to find those, those unknowns. So having, a diverse array of approaches is really important in being able to draw on the interdisciplinary nature of this field. And all of the different perspectives it brings together is hugely valuable. So hopefully, yes, and we'll be able to continue here.
I do like yes and answers from a personal philosophical perspective. I think that with yes and, that's probably the best way to end this panel discussion. I'd like to just immensely thank the panelists. I think that the reason that all the talks were so accessible and interesting and that the discussion was so fascinating was all due to you being very, very interesting people who are good at presenting and have really interesting things that you are working on. So let's thank all of them. And soon-ish, you can talk amongst yourselves again, we will have a discussion actually about one of these controversies that Sarah Ruckheimer brought up, DMS and DMDS, or the lack thereof, on a particular exoplanet. I don't know exactly how that will work, but we'll find out soon.